The overall goal of this procedure is to locally disrupt the blood-brain barrier, allowing passage of molecules into the tissue which are normally confined to the brain vasculature. This is accomplished by first preparing the animal and placing it supine on a water bath above a focused ultrasound transducer and 3-axis transducer positioning system. The second step of the procedure is to acquire MRI images in order to provide targeting information to the positioning system. The third step of the procedure is to prepare and inject the microbubble solution. The final step of the procedure is to set the ultrasound parameters and deliver the ultrasound. Ultimately, results can be obtained that show permeability of the blood-brain barrier at the sonicated locations through contrast-enhanced T1-weighted MRI.
The implications of this technique extend toward therapy of brain and CNS disorders, because this local and repeatable disruption of the blood-brain barrier allows investigation of pharmacological interventions for conditions which previously have had poor response to drug therapy.
Generally, people new to this method will struggle because handling and injecting the bubbles requires patience and care in order to avoid breaking the bubbles before they reach the target.
Demonstrating the animal preparation will be Shauna Rideout, a technician from our lab.
Select an appropriate calibrated ultrasound transducer. Next, place a water bath filled with degassed, deionized water on the bed of a 1.5 Tesla or 3 Tesla MRI. Mount the ultrasound transducer in the tank on an MRI-compatible 3-axis positioning stage or system.